Welcome. We have learned about the growth curve. There are the three types of the growth curve. First one, it is a normal growth curve. Second one, it is a Diauxic growth curve. And the third one, it is a synchronous growth curve. Now here today we are going to learn about the Diauxic growth curve. This Diauxic growth curve, it has been first given by the Jacques Monod. When these scientists they were studying the Diauxic growth curve, they have utilized the two sugars. One, it is a glucose, and this glucose, it is the primary source of the energy, and the second sugar which has been used, it was a lactose. So the first sugar which is used here, it is an glucose, and the second sugar which is used, it is an disaccharide, that is a lactose. This lactose which is a disaccharide, it is made up of the glucose and the galactose. Okay, so it is made up of whom? It is made up of the glucose and galactose. The bond, it is a glycosidic bond. So when the lactose, it has to be utilized by the bacterial cell, it has to broken down the bond between the glucose and galactose, and the enzyme which is responsible to break down this bond, it is called as an beta galactosidase. Means for the utilization of the lactose, the bacterial cell, it has to spend the energy for the synthesis of the enzyme, and that enzyme, it is called as the beta galactosidase. Okay, so here glucose, it is a primary source, that is the whenever a bacterial cell, it is in media, and in the medium, if the carbon source which is present, that carbon source, if it is an glucose with the another carbon sources, then the first reference sugar, it is going to be whom? It is going to be a glucose. After depletion of the glucose, then there will be the thinking of a cell that I will utilize the second carbon source, and here as a Jacques Monod, they have used the lactose. The bacterial cell, it is going to use what? It is going to use the lactose. Instead of the lactose, okay, if here there is a no lactose, then instead of the lactose, if there is a sucrose, or if there is any hydrocarbon, or if there is any lipid, or if there is an amino acid like the tryptophan, then that carbon source, it is going to be utilized after the utilization of whom? After the utilization of the glucose. Okay, so the glucose, it is going to repress the utilization of the another carbon sources, and that effect, it is called as an glucose effect, or it is going to prevent the catabolism of that particular carbohydrate or the carbon sources, then it is going to be called as an catabolite repression. So here you can see, okay, you can see here there is a catabolite repression, there is what? There is a glucose effect, and this, it is given by whom? It is given by the Jacques Monod, by studying the utilization of the glucose and the galactose in the medium. That is here there is a utilization of the two carbon sources. Okay, it has been studied, first glucose and the second one, it is a lactose. Now how it has been studied? Now here the growth curve, okay, the growth curve, it is how there are only the two phases which are alternating to each other, and the first one, it is a lag phase, then there is a log phase, again there is a lag phase, and again there is a log phase. So lag phase, log phase, lag phase, and the log phase, they are alternating to each other. That is, here there are how many phases? There are the two phases. The diauxic, okay, di means what? Two. So here how many phases? The two phases, they are alternating to each other. That's why the growth curve, it is called as an what? Diauxic growth curve. Why? Because the two phases, they are alternating to each other. That's why it is what? It is a diauxic growth curve. Now here on the x-axis, there is what? There is a time, and on y-axis, there is a log of cell number. Okay, so in this way, the growth curve, it has been given. And how it has been studied? It has been studied by studying the mRNA which is present into the bacterial cell. Okay, now here, you know that the lag phase means what? It is the adjustment period of the bacterial cell to the newer environment. Log phase, it is a logarithmic growth phase, it is exponential growth phase where the cell density, it is going to increase. Okay, so here in the first lag phase, what is going to be happen? The bacterial cells, they are going to adjusting to the environment, which is consisting what? Which is consisting of the glucose. So in the first lag phase, okay, the utilization of the lactose, now it has been started. Okay, as the bacterial cells, they are adjusted to the newer environment, what will happen? Now they are going to grow exponentially, so they are going to enter into the phase, and that phase it is called as what? That phase it is called as an log phase. Okay, now as the cell density it is going to increase, what will happen? Carbon source it is going to be utilized. Okay, that is this concentration of the glucose in the medium. Okay, concentration of the glucose in the medium, it is going to be depleted. Okay, so now as there is a depletion, as there is a no carbon source, then the cell what it will think? I have to leave. Then I have to search for what? I have to search for the another type of an carbon source. And now in the medium, what he observes, what the bacteria observes, hey, there is what? There is a presence of whom? There is a presence of the second carbon source, and that second carbon source it is whom? Lactose. But now lactose it is whom? Disaccharide. So how to utilize that disaccharide? Means that disaccharide has to be first what? That disaccharide it has to be first broken down into its subunits, which are there? That are the glucose and galactose. Okay, so here the first simple sugar glucose it will be utilized, and then there will be the utilization of the galactose. But here, okay, when there has to be the breaking down the bond, glycosidic bond between the glucose and galactose, there has to be what? There has to be the synthesis of the beta galactosidase, which is a product of which gene? Lac-Z gene, that we have learned in the lacoperon. Okay, in the lacoperon, there are the three genes, Lac-Z, Lac-Y and the Lac-A genes, which are responsible for utilization of the lactose. In that Lac-Z gene, it is going to code for whom? It is going to code for this beta galactosidase, and this beta galactosidase enzyme, it is allowing the breaking down of the bond between the glucose and galactose, giving the product glucose and galactose. And here this glucose monomer monosaccharide and this galactose monosaccharide, they are going to be formed into the cell. Now here, okay, now in this second adjustment period, which is a lag phase, okay, in this lag phase what is happening? There will be the switch on of the lacoperon. Why? Because what has to be synthesized? Beta galactosidase it has to be synthesized, which is a product of the Lac-Z and which is coming under whom? Lacoperon. So here there will be what? There will be the switch on of the lacoperon into this lag phase. Means what we have, we are saying that lag phase it is an adjustment period. Means what? Here the second carbon source, when it is a lactose, there has to be switch on of the lacoperon. There will be the synthesis of the enzyme beta galactosidase. So for this there is a requirement of some of the time during which there is what? There will be no countable growth it is going to be observed, no detectable growth it is going to be observed. That's why here what you observe, that there is a no growth, that is there is what? There is a lag into the growth, and that's why you are going to observe that there is this second lag it is present. And in this second lag, what is happening? There is a synthesis of the enzymes which are required for the utilization of the second carbon source. Are you getting me? Okay, now beta galactosidase has been synthesized. Now it is going to broke down the bond between the glucose and galactose. Now glucose is available. Okay, now the glucose is available. That's why what will happen? The glucose now it is going to be utilized by the cells. Okay, and again they will enter into what? They will enter into the log phase. Okay, so in this way, when you are providing the two types of the carbon sources, here you can study the sequential utilization of the carbon sources by the bacterial cells, like whom? Like the E. coli or any another bacterial cell. Are you getting? Sequential utilization of the carbon sources. It is studied, okay, it is studied, or it is going to be in a Diauxic growth curve. Okay. So by studying the utilization of the sequential utilization of the carbon sources, Jacques and Monod, they had discovered the lacoperon also. Okay, and also it is going to show that there is a catabolite depression, there is a glucose effect. So until there is no depletion of the glucose from the medium, there will not be utilization of any another carbon sources. Okay, have you got this? So in this way, it is a Diauxic growth curve. I think you may have understood what do you mean by the Diauxic growth curve. If you are like my video, please subscribe my channel and also share my video with your friends, so the knowledge it is going to spread. Thank you.